Hi. Um,
Hi, Sophie.
hi.
This is Heather. Your slides look fantastic.
Thank you. Okay, great. Good to know. I'm split between two screens, so I think this works this way. Okay. So now I can unshare.
Hey, Sophie.
Hi, how are you?
I'm good. How are you doing?
Good, good. It's been a while. Don't remember the last time I saw you in person, maybe 10 years ago.
Probably. Oh, man. Right. So, Nicole, are you also a co-host for this?
I am now,
I just
apparently.
made her one.
Great. Thank you.
Heather, how's stuff looking for OCB this summer?
I am still remaining optimistic. Even if it's a smaller meeting, it's either in person or it's not happening.
Yeah, I will be
I'm
there.
not doing a virtual meeting again.
I will be in person. And actually, I was talking with Sophie Clayton the other day, who's also for the OOI meeting beforehand, and I was like,
Yes.
we have to do like a group happy hour or something beforehand. We got to get people together.
I love it. I think we should do it. Actually, oh no, the tent won't go up for OCB until the weekend, like the Saturday or Sunday before. I was going to say we could get takeout and hang out in the tent.
Oh, that'd be so
we
good.
can get takeout and go to the beach. What am I thinking?
Right? Yeah. Just take out and go literally somewhere.
Mm-hmm.
Sunset at Chappie with a few pizzas and a cooler of beer.
Ooh, let's just take over the knob.
Yeah.
That's kind of a long way to haul beer.
I don't care.
All right. So, Sophie, are you okay with us recording your presentation?
Yeah, for sure.
Okay. Because we have been doing that for people who then aren't able to attend today so that they can watch it later.
Great. And the discussions will also be recorded afterwards?
Have we been, we haven't,
So we don't record. Yeah, if we do breakouts, we can't really record those. But if we have full group, usually we're recording anything that's happening together in full group, whether it be presentations or discussion.
okay. So we come back in after the breakout groups for about 20 minutes. Is that right, Nicole?
Yeah. Because we usually do like go to breakout groups, come back, and then go back to breakout groups.
Oh, we have 2 breakout groups?
We have enough time. It depends because how much time it takes because we've got 90 minutes. And so if it's done right, I shouldn't say done right, but if we're able to, we just like, about 30 minutes for presentations. And then if we do 20-minute breakout group, come back for 10 minutes and then do another 20-minute breakout group and then 10 minutes. That brings us to the whole hour, right?
And do you want random breakouts? Like just break people up randomly? And what size are you shooting for so I can gauge how many rooms to open?
That will depend upon because we should have, we've only got 3 that can actually host a room today because Karen can't make it and Jessica's not feeling up to hosting a room. So it's just gonna have to break us into 3 groups.
Okay.
Okay, so 3 rooms.
It may not move you as co-hosts. You might have to select on your own.
Okay.
Because I have you as co-hosts, but
okay.
That's good because you can kind of decide, oh, I'm going to take 1, I'll take 2, I'll take 3, and then sort them out now.
That's good.
Becky, do we already have a Google Doc and like for notes for this?
Yeah, we do, and it's in the OCB working group folder, and it says February 2022.
Got it.
I'm gonna open that too. Yeah.
Yeah.
Thank you. I'll make sure to paste that link in.
Okay, great.
All right. I gotta ask, is there any way to change my name? I accidentally did change it from my email.
I'm
trying to figure it out here.
You are probably the only one that can do it. Try right
Yeah.
clicking on your name. Oh, thanks.
Video setting. Not the most important thing. All right, I'll just leave it. It's fine.
Nicole, when I tap the participants tab, when I mouse over my name, there's a little, almost like an ID card, and that allows me to edit my profile.
Okay. And
Okay, and Sophie, you're still a postdoc at the what is it? The American Museum of Natural History?
Yes. Yes,
And
that's still
you're
right.
okay, and you're working with Yoon Soo Kim?
Insu Kim, yeah.
Okay.
are you in New York right now?
Yes, it's a cold day. And it turns out it's the day where my radiator stops working. So it's kind of
No.
cold.
So unfortunately, my internet does not necessarily behave well.
So I'll
Are
be
you
the
still
primary host, so I don't think you shouldn't have to worry.
Okay.
You're a co-host, but I don't think anything's gonna tank, so don't worry. I say that now.
Except,
I'm gonna knock on some
except
wood
my
now.
internet.
Yeah, if your internet tanks, we'll know if you disappear.
Yes, so I don't know where Suzanne is.
Yes.
Saw her last week, and I know is. She was like, "See you Monday." So
She knew what day it was.
Yeah.
Heather, will OCB be at OSM at all? Are you guys gonna have an exhibition?
No, we're not doing an exhibit booth. We're doing a couple of town hall meetings, and there are a lot of our working groups are having sessions, but we're not doing an exhibit booth. We didn't think it would be worth it to do any more virtual. I'm just holding out for the next one in person.
Well, I just emailed Suzanne to see if
Is having any issues?
I don't know. Oh, there she is.
Yay.
Hey.
Susana, would you like to try sharing your screen?
Oh, should I go first?
I just wanted to let you practice sharing your screen and make sure you're comfortable with it so that when it's time to give your talk, you would
Yes, thank
have
you.
peace of mind.
Thank you.
I've just made you the presenter.
Okay. Do you see my full screen?
Mm-hmm. Looks good.
Yep, great.
Now, who is going first today? I'm forgetting. Is it Sophie and then Susanna, or is it the other way around?
I thought that's what it was, but I don't.
It's
It's fine by me.
okay.
I think you've been listing Sophie's name first, so we can
Right.
stick with that.
That works.
Sophie, you're now the presenter again.
And Sophie, is it Charvet or Charvet? I don't remember.
Yeah, it's French, so it's Charvet.
Charvet. Okay, great.
It's 11 o'clock right now. I don't know if you want to give it another minute.
Probably
Still
we've got
got
some people.
people still
Presented.
wandering in.
The screens keep getting smaller. Okay, I think maybe we can go ahead and start. So thank you everyone for joining us today for our first workshop session in 2022. Today we're going to have two speakers, Sophie Charvet and Susanne Wilken. Sophie comes to us from I have to keep looking this up, the American Museum of Natural History, where she is a postdoc with Yinsu Kim. Sophie got her P from the University of Laval. And she is going to be our first presenter today.
Thank you, Rebecca. So let me share my screen here. Yeah, so I'm going to be presenting some data from a study that was done in collaboration with the Lamont Doherty Earth Observatory at Columbia University, also here in New York, where we use transcriptomics to explore mixotrophy in green algae. In green algae are close parents to land plants. We find them amongst the streptophytes and the chlorophytes. And so as such, they are generally characterized as strict photoautotrophs. But there's an increasing number of studies that are starting to show that bacteriovory might actually be quite common in some green algae, particularly among the prasinophytes, these basal branching lineages that are actually quite diverse. So in a previous study, we used the gene-based model to predict the mixotrophic potential of all green algae for which there is publicly available genomes and transcriptomes, such as these. This model was published by Burns et al. In 2018, and so John Burns' model is based on the intersection of probability scores indicative of the presence of 3 types of protein sets: proteins that are enriched amongst photosynthetic organisms, proteins enriched amongst prototrophs, so plants, photosynthetic algae, and fungi, and protein sets enriched amongst phagocytes. So predictions for each of these 3 categories can then provide insights as to the actual trophic mode of the organism. So the scores can then be projected onto the small wide representation, and organisms that land in the green section are predicted to be strict autotrophs, such as was the case for most of the Chlorophyta and Streptophyta in our study. And then organisms that land in the blue section are predicted to be phagomixotrophs with high scores for photosynthesis and prototrophy, but also high scores for phagocytosis. And this was the case for quite a few percentophytes. So our next step was to further empirically test these predictions by using strains we had in the lab. So we confirmed bacteriovory for 5 strains using microscopy and flow cytometry. And on top of confirming their capacity to ingest bacteria, we also found that most of these strains displayed an increased intensity of feeding activity under nutrient limitation, shown in the right panel here. So we decided to explore that by comparing gene expression between the nutrient replete medium and 2 forms of nutrient restriction. So these nutrient restriction experiments were done by focusing primarily on 1 strain, Pterosperma cristatum. So we exposed Pterosperma to 2 different growth medium. F/2 is the standard growth medium for marine phytoplankton and contains all nutrients at replete concentrations. And F/20, which is the diluted version of that, is therefore a general nutrient restricted culture. We then monitored growth of Pterosperma over 16 days and saw that both cultures grew similarly for the first 16 days. And after that, the nutrient restricted culture showed a slowdown of growth, potentially crashing, but since we stopped sampling at that time, I can't speculate as to that. But during this growth curve, we also collected samples for nutrient concentrations at day 0, 11, and 16. And this showed that while F/2 cultures remain nutrient replete throughout the experiment, in F/20, the nitrate concentrations, as represented here, were below detection threshold already at day 11. So by day 16, Pterosperma had been growing in nutrient depleted conditions, essentially, for at least 5 days. And this was also the case for phosphate concentrations that we looked at. Now, we then looked at feeding rates that are represented here as the percent fed Pterosperma cells per minute. And so these feeding rates remained stable in F/2 cultures, and actually none of these were ever significantly different from that of the negative controls that are represented in gray. In contrast, the F/20 culture showed an increasingly an increase in feeding behavior over time with significant differences from the negative controls at day 16 and day 11. And so this difference in feeding behavior leads us to wonder whether Pterosperma is using bacteria as a source of nutrients to maintain its photosynthetic growth in this nutrient-restricted culture. And if this was the case, then first of all, we'd see an increase in phagotrophic processes, and we would see a maintenance of photosynthetic processes in the transcriptomes. So to carry out these comparative transcriptomics, I collected RNA from at 2 different time points, but from Pterosperma in 3 distinct physiological states relative to nutrient availability and feeding behavior. So there was the replete control, which is the reference transcriptome, to which I then compared the nutrient-restricted treatments, i.e. the limited state and the depleted state. So I did pairwise comparisons of each nutrient-restricted treatment against the replete reference to obtain a list of differentially expressed genes. And then each significantly differentially expressed genes can be plotted as the log-fold change of the first comparison on the x-axis against the log-fold change from the second comparison on the y-axis. So looking at these log-fold change scatter plots, the quadrant into which a gene is plotted will provide information as to its relative expression level compared to the reference transcriptome. So for example, genes plotted in quadrant C are down expressed in both nutrient restrictions, while genes plotted in quadrant B are up expressed in both nutrient restrictions. And then if they're plotted in quadrant D, they will only be up expressed in the limited state. And if they're plotted in quadrant A, these genes are only up expressed in the absence of nutrients or in the depleted state. So looking first into processes potentially involved in phagotrophic activities, we can see that there's a shared upregulation of 1.5% of the autophagy-related genes. And similarly, for the genes involved in endocytosis or phagosome formation and transport, 3.2% of the genes involved in these pathways were upexpressed, which represented about 31% of all differentially expressed genes for that pathway. In terms of lysosome-related genes, such as proteases and lipases, and so digestion enzymes, some were upexpressed, some were downexpressed in nutrient restrictions, and there is no clear trend in this situation, so it's a gene-by-gene basis that we would need to dig more into. And then we saw a significant upexpression of genes involved in peroxisome-related activities, either under both nutrient restrictions or solely under the depleted state. So our first conclusion is that most of the differentially expressed genes involved in phagotrophic processes were significantly upexpressed under nutrient restrictions. So there is an increase in phagotrophic activity, which reflects the increase in bacteriovory that we observed in our cultures. Now, looking into phototrophic related genes, we find that almost half of all genes involved in light reactions of photosynthesis, so antenna proteins, the complexes photosystem 1, photosynthesis 2, and any protein involved in electron transfer chain are down expressed in both nutrient restrictions. And this was also the case for genes involved in terpenoid metabolism, such as the biosynthesis of chlorophyll and carotenoid precursors. Genes encoding enzymes from the Calvin-Benson cycle involved in carbon fixation were also, for the most part, down expressed in nutrient restrictions. And this was also the case for starch metabolism, so genes involved in starch synthesis, a storage compound, a carbon storage compound unique to photosynthetic organisms. So as a second conclusion, the phototrophic activity actually appeared to be downregulated under nutrient restrictions. So having a quick look next at the heterotrophic pathways that could be detected in our transcriptomes, such as oxidative phosphorylation, which is the respiratory electron transport chain. We can see that although there was a limited number of genes from that pathway that were differentially expressed, half of them were downregulated or down expressed. Similarly, for the TCA cycle, most genes that were differentially expressed were down expressed in both nutrient restrictions, except for 3 key enzymes that were solely upexpressed in the depleted state, such as malic dehydrogenase and citrate synthase. So key genes are being modulated between these treatments. And then in terms of glycolysis or gluconeogenesis, we could see that the majority of the genes in that pathway were actually down expressed, with just key genes being up expressed in either the limited state or the depleted state. And it turns out that these are actually rate limiting enzymes that tend to increase glycolysis activity in the cells. And the pentose phosphate pathway is another alternative to glycolysis pathways. And similarly, we had key genes being mostly downregulated under nutrient restrictions. So heterotrophic pathways were differentially impacted by nutrient restrictions, and the actual output that results from these differential expressions isn't always very clear, and so we need to dig deeper on the gene by gene basis to see exactly what activities this might reflect. But if we go back to our initial question, which was whether or not pterosperma is using bacteria possibly as a source of nutrients to maintain photosynthetic growth, regarding the first hypotheses where we saw where we expected to see an increase in phagotrophic processes. This seems to be corroborated by observations where we saw an increase in the expression of genes involved in phagotrophic activities, such as ingestion and digestion. However, the second hypothesis regarding the maintenance of photosynthetic processes actually seems to be contradicted here by our observations, where we saw an increase in the expression of, a decrease, critical word, a decrease in the expression of the photosynthesis genes and the carbon fixation genes. So it appears that photosynthesis was actually not maintained in pterosperma under nutrient restriction. And this suggests that either the bacterial concentrations were too low to sustain photosynthetic activity in these particular cultures, or that as a general tool, prey are not used for nutrient supplementation by pterosperma. And in fact, our observation of this modulation of heterotrophic processes, such as the TCA cycle and glycolytic pathways, seems to indicate that pterosperma might be increasing catabolic or anapyrotic activities to obtain energy from carbohydrates, possibly obtained from its prey, instead of photosynthesis for growth. So these results provide us with new working hypotheses to test in the future with more physiological or biochemical experiments. Biochemical experiments. And thank you all for your attention.
Thank you very much, Sophie. We have time for one or two questions if anyone has any. Oh, Holly?
Thanks so much, Sophie. That was a great talk and also really beautifully and clearly illustrated. I was super curious about your comment that perhaps so let me back up for a second. So you observed the same growth rates in the nutrient deplete and nutrient replete cultures, if I recall correctly. So does that imply that if these organisms are switching to bacterivory, that sort of perfectly substitutes in terms of growth? I'm curious about what your thoughts are about what might be setting the growth rate for these cultures and why it's so similar across treatments.
Yeah, it's tricky. It varies. So if we repeat these experiments 4 times, 5 times, there's a variability here between experiments. But yes, it's interesting to note that growth is sustained even when we cut down so far on the whole range of nutrients, right? We haven't isolated specifically whether it's nitrate or phosphate or vitamins that are in the medium that are so essential, but in any case, pterosperma seems to be able to supplement for any of those in this situation. We didn't we didn't control for the amount of bacteria that it was growing with. There was an ambient community that we quantified, but we didn't try to alter or interfere with any of that. And then we worked with fluorescently labeled bacteria to estimate feeding rates. So there might be a difference in growth if we provided more bacteria or less. And that would be really interesting to see.
Thank you.
Great. And one more question from Isat, or if I've said that correctly.
Close enough.
Okay.
Question is, how does light value, not amount of light, but changes in the wavelength, do you think would affect this process? That is, if we go from photosynthetically active light to less photosynthetically active light, how is that going to alter the, considering that question, I'm asking the question because of the consideration for depth. Water.
Yeah. So we did similar experiments where we tried to limit the amount of light using filters, but they were neutral filters. So we weren't taking out different wavelengths. We did see overall less growth and less feeding, more feeding than compared to the non-restricted cultures, but less feeding compared to the nutrient-limited cultures. So light is completely a different influence on pterosperma in particular. I don't know how it would respond to filtering out certain wavelengths as this would happen in the water column. But given its reaction to the overall reduction of light, it seems to be reacting more strongly to nutrient restriction than to light restriction. So I think it'd be similar if we took out specific wavelengths.
Thank you.
Thank you very much. All right, we'll move right along to our next speaker, Susanne Wilken. She is coming to us from the, wait a second, University of Amsterdam in the Netherlands. So she's on a different time zone than we are.
Yes.
And where she is an assistant professor in aquatic microbial ecology.
Yes, thank you very much. So, of course, the session is supposed to focus on omics techniques and their use to study mixotrophs. And I think the most challenging part of that, as Sophie already partly illustrated, is to actually identify the signal of phagocytosis in phytoplankton, because we know a lot about the photosynthesis and nutrient uptake, but phagocytosis is much less well studied. Now, I like to illustrate phagocytosis in a very simple way, as you can see here, just a very simple cartoon of a cell that has chloroplasts and can feed. But of course, you're all aware that it is actually a much more complex process. And that is here just one illustration of the process of phagocytosis taken from an immunology paper, as a lot of the knowledge that we have about that process comes from the field of immunology. And you can see that, well, of course, the cell needs to ingest the bacteria. You can even imagine that prior to being able to ingest prey, a protist would need to express some form of motility to find prey, express receptors to actually detect the prey, and to then start the cytoskeleton rearrangement that lead to the ingestion of the prey. And once a food vacuole is formed, it is altered through several merging of different vacuoles. The pH is reduced by these ATPases. And then finally, of course, digestive enzymes are needed to actually digest the prey. Now, this is a pretty, the process of phagocytosis is an ancient eukaryotic trait, and it's very complex, so there's hundreds of genes involved. But the difficulty is that many of these genes are not necessarily unique to the process, but might also be involved in very different processes. So it's really difficult to see what we are actually looking at. Now this a little bit as an introduction to phagocytosis, but now I want to get to the two main points that I want to talk about today. And the first part is phagotrophy in bloom-forming haptophytes. This is work that has been led mainly by the PhD student Sebastian Capelle, who I think he is also in the audience today. And then I will move on to transcriptome experiments focusing on phagotrophy in the chrysophyte Ochromonas. But first, the haptophytes. So this is a study that is based on field work in the marine systems that are closest to where our university is situated in Amsterdam, so the North Sea. And we were not necessarily meaning to target haptophytes, but we wanted to, to target a season where we would find mixotrophs in the natural community. And that is most likely to happen in late spring when the nutrients are depleted by the spring bloom. Now, another phenomenon that often occurs in this area in spring is maybe something that some of you are familiar with. We often have these huge foam events where the foam washes on shore. And that is actually caused by a bloom-forming haptophyte, Phagocytosis globosa. So not necessarily intended, but when we actually sampled the spring community, we happened to be at the peak of a Phagocytosis bloom. Now we performed a series of incubation experiments, but I will not go into details of those, but only focus on one aspect of this work, where we tried to look at feeding in the mixotrophs that were present, which happened to be Phagocytosis. And we used a double cardfish approach for that. So we used a mix of different bacterial probes to represent a large fraction of the naturally occurring bacteria, and those are labeled in green. And because Phagocytosis was the most dominant phytoplankton in this situation, we then also combined these bacterial probes with probes that target Phagocytosis. And that you see in red in the figure. And then in blue, you can see faintly the nucleus, and you also see a faint red background, which is the remaining fluorescence of the chloroplasts, which are mainly bleached by the staining, by the cardfish protocol. Now, from this image on its own, it's hard to actually see whether these bacteria are maybe externally attached or whether they have really been ingested by Phagocytosis. But Sebastian spent many, many hours at the confocal microscope, and that is what you see here. So then we get basically a scan through individual cells of Phagocytosis, looking at different focal planes. And you can see here that there is bacterial cells that are really inside Phagocytosis globosa. It almost looks like they are enclosed in little vacuoles. So that, of course, we were excited about that. So we thought maybe Phagocytosis is actually feeding on bacteria. From this observation alone, it's hard to really conclude that, because we did, of course, not add any tracers, but it is just labeling the natural bacterial community. So we took a second step, which is actually very similar to what Sophie just introduced. So we used the gene-based predictive model published by John Burns, which Sophie nicely introduced, so I will not say much about this. And we used it for publicly available genomes and transcriptomes of haptophytes to predict their trophic mode. And not surprisingly, pretty much all of them were predicted to be photosynthetic. We, of course, know that they are photosynthetic. And also, most of them, or many of them, are predicted to be capable of phagocytosis. And also this, of course, doesn't come as a surprise, because we know that there's many mixotrophs within the haptophytes. But the group that we were most interested in are the Phagocytalis. So those are bloom-forming haptophytes that have not been observed to feed before. And even within those, we see quite consistently a prediction of phagocytosis capability, but there's quite some strain-to-strain variability as well. So there might be some strains that do and some strains that do not feed. Now I will leave it at this, but I think that this finding is quite interesting because it could be another group to zoom into more and then to carefully experimentally validate whether some of them are feeding and others are not feeding. And it could be a very interesting group to study, well, basically the disappearance of phagocytosis in a specific group of phytoplankton. But one notion that also John showed in his paper, and I think many of you are aware of that predicting phagocytotic capability is of course very different from predicting the actual activity of the process. And especially in these haptophytes, we know that most likely, most of the time, they might not be feeding. They might only be feeding once in a while. So how do we actually know what they are doing? And this brings me to the second part of my presentation, which is phagotrophy in the chrysophyte Ochromonas, and the question whether we can trace the phagotrophic activity in transcriptional signatures. Now, this work has already been done quite a while ago. It painfully reminded me when preparing this presentation that I would have loved to long publish this, because it's work that I did as a postdoc in Alexander Gordon's lab at Ambari. So first, the model organism that I used, Ochromonas, is an obligate mixotroph. You can see here its growth rate either only in the light when not being fed at all, and these are azenic cultures, so they do not have any bacteria available and they do not grow under these conditions. Then you can see that when they have both light and prey available, they do grow reasonably well. But also in darkness, they are not able to grow. So they are really obligate mixotrophs. Now, when performing an experiment with an obligate mixotroph, obviously I need to feed it. And I used fluorescently labeled bacteria here. You can see well, mixotrophic conditions. So at least under the microscope or in a flow cytometer, we see these mixotrophs as having both red fluorescence from their chloroplasts and the green fluorescence from the prey that they are feeding on. Now, in order to manipulate their actual feeding activity, I then introduced a second treatment where I let them deplete their prey. And I could do that for about 3 days because I did not want to have them stop growing, of course. Then we would not have a meaningful transcriptomic comparison anymore. So I let them deplete their prey for about 3 days. And after these 3 days, I then started a third treatment where I added prey back to the treatment that had previously depleted their prey. So then at the start of my transcriptome experiment, I have 3 different treatments. And what you see here is how these treatments behave over the course of a 24-hour experiment. And maybe on the top, you can see it first. So the treatment that has depleted the prey has the lowest green fluorescence. So on the y-axis, you see the green fluorescence that is indicative of the prey content of the individual cells in the flow cytometer. And the treatment that has continuously been fed with prey has, of course, a higher green fluorescence. And then you can also see that the in-between treatment that received prey back after having depleted it started off with low green fluorescence and then over the course of the experiment increased in fluorescence and is more comparable to the other treatment that has been continuously grown under mixotrophic conditions. Now, the lower panel here is just to show you that when we are looking at a short-term experiment, we need to actually take into account when the cells divide. So here you see growth plotted as divisions since the start of the experiment, so divisions since T0. And basically what you see is that everything stays flat during the light period and the cells start dividing during the night. And they might be slightly delayed. The different treatments have been slightly delayed in when they start dividing. And for that reason, we then chose to actually sequence the transcriptomes only within the light period. So we do not get the signal of of differences in cell division. Now, in total, we had a pretty high number of differentially expressed genes. More than 4,500 genes were differentially expressed between at least either 2 treatments or 2 time points. But the multidimensional scaling that you see here actually indicated that they behaved roughly as we expected. So we have the treatment that depleted the prey, very similar to the one that just received the prey at the start of the experiment, while the mixotrophic treatment, of course, was different. And then we see that over time, this in-between treatment switched after having received prey quite quickly to be more similar to the mixotrophic treatment. And you can also see that there is a very clear pattern of time. So the timing of the day really matters when we look at the transcriptome. Now then we can analyze the different patterns that we see by clustering the differentially expressed genes based on their up or down regulation in different time points and treatments. And that led us to 4 main clusters. And here you see the first 2 of them. Now we have on the x-axis the 3 different treatments. And within each of those treatments, the 3 different time points. And on the y-axis, the up or down regulation as a log2 fold change. And basically what you see, these 2 clusters are those genes that mainly show a temporal pattern of up or down regulation. And this might not be too surprising. So as I mentioned, we have all these 3 time points are still within the light period. So we started early in the day and then we ended later in the day. And over this time period, genes involved in light harvesting and translational termination, and also in chlorophyll biosynthesis, were downregulated. So apparently, genes involved in photosynthesis were downregulated towards the end of the day, which might not be surprising. And then the genes that were upregulated were enriched in genes involved in DNA replication and repair, and also in mitotic chromosome condensation. So also this might not be surprising, having in mind that the cells are getting ready to divide at night. So we see a strong signal that does not necessarily have to do much with their feeding activity. But then the next 2 clusters are the ones that responded more to our treatment. So here you see genes that are either up or down regulated when there's no prey available, and they show an opposite trend when there is prey available, and these in-between treatment that received the prey at the start of the experiment kind of shows a switch between these 2 conditions. And what we see is that the genes that are upregulated under mixotrophic growth are enriched in genes involved in translation and transcription. So this might mainly be a reflection of better growth conditions. So as I said, this mixotroph really requires the prey to grow. So once they get prey available, they might just reach higher growth rates. And other functions that might be a little bit more interesting are, for example, peptidoglycan catabolism and proteolysis, which might actually reflect digestion of prey in the process of phagocytosis. So this could actually be a signature of phagocytosis. And then genes that were downregulated under mixotrophic conditions were involved in cell motility, in calcium transport, and in signal transduction. And this is a little bit harder to interpret, maybe. But what I was initially a little bit surprised or maybe disappointed was that, while we don't really, looking at the data like this, we don't really see a clear signal of phagocytosis. But then I looked at the same data in a different way, and I basically picked out those genes that, based on literature references, were known to have some role in the process of feeding. And I categorized those in the time frame in which they are required to feed. So this involves genes involved in motility. So this is supposed to reflect a flagellum where you have tubulin filaments and motor proteins that move around those. Then the actual process of ingestion, where a prey is detected by receptors, and there's several signaling components that are required to then trigger a change in the actin cytoskeleton that actually leads to the ingestion of prey. And then there is components that are involved in phagosome maturation and in the actual digestion of prey in the phagolysosome, like, for example, digestive enzymes and transporters. And if I then map all the differentially expressed genes that are likely to be involved in phagocytosis, and here again we have those three treatments, the mixotrophs that are hungry, basically, that have depleted their prey, the ones that are feeding, and the ones that have been fed in the course of this 24 hours. And I think it's quite striking that then we suddenly see a relatively clear pattern that components that are involved in feeding in the early steps of phagocytosis are actually upregulated in the hungry cells. So these are processes involved in motility and in the detection and ingestion of prey. So to me, that almost reads like the hungry cells are getting ready to find and ingest prey. On the other hand, in the mixotrophs that are actually feeding, and especially in the ones that have just received prey, the steps that are needed later on in phagocytosis seem to be upregulated, and in particular, components involved in digesting prey. Yes, so this brings me already to my last slide. So I think based on this, we can actually see distinct transcriptional patterns in actively feeding versus hungry cells. And those somewhat reflect the temporal sequence in which those are involved in the process of phagocytosis. And of course, I have the hope that this could help to, in the future, identify nutritional states or the condition that the cells are in from, for example, metatranscriptomes. But on the other hand, it's really hard to already conclude how general these responses might be. Obviously, as we've also heard from Sophie's talk, that different mixotrophs might feed for different reasons and under different conditions. So there's probably, this functional diversity is probably reflected also in quite a diversity in transcriptional patterns that we see. And with this, I would like to thank you for your attention and also thank all of my co-authors.
Thank you very much, Susanne. We'll take a little time for one or two questions if anyone has them. No hands are popping up. Oh, Natalie Cohen.
Hey, Susanna, I really enjoyed that talk. I thought that was really lovely work. Well, I guess I have a few questions, but the data that you just showed with the heat map of the genes involved in motility, that was so striking. And I guess just to play devil's advocate, I'm just wondering if you think some of those genes might be involved in a general stress response. And I ask because we see some of them in the deep ocean, and although I think it could be involved in phagotrophy, what are the chances that that's just what cells look like when they're growing poorly and they don't have nutrients and they're struggling? Just curious if you thought about that.
Yeah, I think I agree that it's really hard to know what they are involved in, right? That's yeah, I can't quite answer that. So it's hard to really be sure that it's a signal of phagocytosis. But it is, of course, in this condition, the one thing that we alter the most. But in the obligate mixotroph, it's impossible to alter feeding completely independently of growth state. So that is hard to say. But which do you refer to particular genes that you see up or down regulated in the deep sea? I'm wondering.
Yeah. one of them are the cytoskeletal proteins like tubulin and actin, which could be involved in motility, but as you know, they're probably involved in a lot of different processes as well.
Yes.
So those are ones that I think about a lot.
Yeah.
Okay, I'll let other people ask questions if they have any.
Sorry, my video is just glitching. So if anybody had a question. Oh,
I don't see any questions right now.
okay, so let's go ahead and go to our planned breakout groups. We'll be discussing a couple of questions, at least in our first breakout group session for about 20 minutes. There will be a recorder associated with each of those. And after the 20 minutes, we'll come back to the big group and discuss a little bit what people have talked about, and then break out again. We can either continue discussing those questions, or people can start to raise other issues that they've thought about that go beyond those. And then we'll come back again to discuss that. And I think that Heather is going to just randomly assign us to breakups.
Yes, I am doing that, finishing that up here. Okay, so if anybody wants to move rooms, you can easily do that, so please feel free. I've tried to make these even.
All right, Heather, do we have everybody back?
I believe we do.
All right. So my video and internet keep glitching, so I will ask somebody else from my group to present the information that we have. We were in breakout group 1. Sophie, would you be willing to do that?
Yes, yes. Let me
Thank you very
find
much.
document. Okay. So with regards to the first point, which were regarding strategies to identify patterns that are descriptive of in-situ nutritional balance, our group came up with the idea that we need more available databases for annotation that would be specific to protists and to mixotrophic protists. We need a standard way of analyzing transcriptomes for microbial eukaryotes in terms of number of replicates necessary, the phase of the growth during which we sample for transcriptomes, and so on. So a standardized approach at the lab and experimental level, but also a standardized approach at the bioinformatics level. So in terms of annotation of the processes that we're interested in, yes, emphasis on the point of the growth phase of the cell cycle at which the transcriptome is sampled, looking into environmental impacts. Ooh, can someone help me out here?
I think that was Isat's point about understanding the environment, and
oh, yeah. So how that also influences the cell cycle growth phase or any noise that would be in the transcriptome. So stress factors that are not necessarily related to the mixotrophic process that we are targeting. And where the energy is supposed to come from to help understand that environmental response. And then get baselines for different cell stages or activities. So have a sort of database of baseline transcriptomes to which we can compare and contrast the variable transcriptomes to study different processes. To the second point on how to utilize or apply this information for in situ studies of mixotrophic activity. We refer to metagenome bins, functional, but also as much as phylogenetic binning of the different genes, and have to discuss which gene sets to focus on. I think with regards to relevant protein sets for phagocytosis or photosynthesis or prototrophy, for example. And then the third point with regards to relevance of establishing model organisms, there's a big yes, everyone wants and feels the need for having model organisms to work with in the lab. So we would need to make decisions on the taxonomy that would be represented in these model organisms, and decide on this as a community. In terms, so yes, the choice for these model organisms would be based on the taxonomy, but also based on making sure that there's a proper representation of the functional diversity of mixotrophs among these model organisms. And the use of model organisms, for example, for knockout experiments or to test or using inhibitors to block certain processes in order to investigate the importance of these processes for these organisms to study functional importance. And I think that was it.
Thank you very much, Sophie. I realized I threw that at you without much warning. You did a great job. Thanks. Breakout group 2.
Sure. So I'll report for Breakout Group 2. Thanks, Sophie. I think you teed us up really nicely there. We also talked about the need for good databases, and we saw the Burns one highlighted here. There's also one by Ben Lambert built on MMETSP data. But there's also this challenge that when we do metatranscriptome work, we sometimes see that half the data can't be assigned. So clearly some ways to go there. We spent a fair amount of time talking about how to potentially use this type of information for in situ studies. So we talked about the challenges of phagotrophy in particular being sort of this ancestral pathway. So needing to figure out where genes fell in which pathways and disentangle that as being a challenge to actually going from gene expression to function. We discussed whether single cell work could be of value here, because that would allow you to sort of circumvent the problem of having a bulk metatranscriptome and asking which genes came from whom. And perhaps being able to do a transcriptome in a single cell is within our scientific lifetimes, perhaps a tangible goal in the next few years. And this could be really valuable because it could give us a handle on what sorts of plasticity these mixotrophs exhibit, the ability to kind of detect that in situ on samples that you've just taken from the field. And that brought us also to thinking about the importance of model organisms as a way to benchmark some of these approaches. And we talked about the need for having diverse model organisms from diverse lineages that show a range of plasticity, a range of reliance on phagotrophy versus phototrophy, so that we could sort of try to constrain what gene expression measured in field settings might mean in terms of function. And we also talked a bit about how one could determine absolute number of transcripts in field samples and then try to map that to any rate measurements from field data to be able to connect omics approaches to biogeochemical function. Group 2, did I miss anything? I'm sure I did, but it's the gist. Thanks.
All right, moving on to group 3. So we started with the importance of establishing model organisms. Several people in our group felt very strongly that this needs to happen. Really, the question there is how can you possibly interpret in situ data without having some sort of reference? So we talked a lot about that and what makes a good model organism. So some of the things that we thought would define a good model organism are, can they represent a broad range of modalities or survival techniques that mixotrophs have? Are they prevalent in the ocean? One thing also is, do they have attributes to other fields? So are they studied in other labs for reasons unrelated to mixotrophy, but that could be useful for us. And so some of the thoughts that we had on model organisms, let's see. Right, we need as much data as possible. So that's one of the reasons why we want it to be studied in other labs, so that, you know, it's a hard work. And we kind of estimated that there's probably 5 model organisms in general, 10 model organisms generally that we use in mixotrophy studies, and maybe 5 that can be specific to omics studies. So we started there and then moved to strategies for omics techniques, and we kind of combined questions 1 and 2, but basically, our strategy so far has been single species under tightly controlled conditions as a very important first approach. So how do we move into a natural community and manipulate natural community, which is clearly the next needed step? Comparing physiological activity across natural gradients to transcriptome data, and then how do we take this data and incorporate it into a mechanistic model with rates? And so looking at nutrient uptake measurements to compare with transcriptome data and activity, upregulation and downregulation. Of course, it's going to be very hard to get good enough relationship to develop conversions. yeah, so I think we've all kind of hit on the same questions and want to highlight like the need for many more model organisms. Did I miss anything, group 3?
Thank you very much, everyone. That was great. We have a lot of good notes from this, and I appreciate that you guys have taken the time to join us. We're coming up on 12:30, and I'm going to suggest that we finish up here rather than having another round, unless people really want to stay for another 20 minutes, which I think we'll probably all need to get going. So I really appreciate that you joined and that you took part in a great discussion. And I want to thank again Sophie and Susanne for their talks. They were awesome. And I am really so happy to see that work on Phagocytosis because it keeps coming up in our BRDU libraries. And we're like, oh man, is it really eating?
Yeah, I should have mentioned that. Yes, I'm aware of that.
Yeah. So yeah, it's that's great to see. So thank you very much, everyone. If you have any, oh, sorry, go ahead, Nicole, you have your hand up.
Yeah, I just wanted to say one last thing. Anyone attending Ocean Sciences, we have an event for this group on Monday, the first Monday of Ocean Sciences, February 28th at, we start at 10:30. I think, no, is it 11:30? I think it's 11:30 to 1:30 is when we have 2 hours of oral talks and discussion, and then our posters are combined with another group, and they are spread out on Monday evening and Tuesday evening. Find those sessions. So some really cool talks, it looks like, from people who haven't even been here at these ones. So hearing from other people who are doing mixotrophy research, it'll be worthwhile to stop by.
Nicole, is that EST or CST?
That is ME. You mean, oh no, sorry, the time zone. EST. All the times for Ocean Sciences are put in EST.
I thought they were in CST. Okay, thanks.
Thank you, Nicole, for reminding us about that. Heather has posted in the chat that our next working group meeting will be on April 11th from 11 to 12:30, and the theme is biogeography. The speakers have not been determined yet, so keep your eye out for that. We'll be posting and sending out reminders for it. All right, everyone. Thank you very much. Have a great afternoon. See you at Ocean Sciences.
Bye.
Bye-bye.
Thanks, everyone.
Have fun.